What I'm going to talk about today, it's kind of a collection of projects that we've worked on, a few of us from the Murray-Darling Freshwater Research Centre, where we had a small collection of projects working in and around the Broken Creek. And as Leanne has mentioned, we've rejoined sort of the task force who are actually doing some work in that area. So what I thought I would do is give it a little bit of a history of the Broken Creek, then talk about some of the issues, where and when and why MDFRC got involved with the Broken Creek and what we were trying to do, and then I guess just talk a little bit about the newish project that we've actually just started, and then I guess at the end consider maybe roughly what this water body is like and what the sort of future holds for it as a creek and a water body of ecological significance. So that's roughly what I'm going to do. So the project team's growing, and as I said, it's the project that I'm going to talk about is actually a collection of projects that have involved a few people over the years. The original project team was myself and Darren, and we were more than ably helped out. In fact, the folk who did a fair chunk of the work, Karina, Shane Perryman, Jess Wilson and Garth Watson were involved with that. The original work was funded by the Golden Broken CMA, GBCMA, and they were particularly interested in what was going on in the Broken Creek. But more recently, the new work is actually being funded by the Commonwealth Environmental Water Office. So that's the new, but it's also a little bit of the history. So what are we talking about? Where is the Broken Creek for those who don't know? And I'll try to point out with the pointy thing here. Basically, the Broken River, I've just focused in on the creek up there. The Broken River is running from bottom of the screen here through Benalla and swings out up into the Goulburn. Broken Creek actually comes off the Broken River at a place, Casey's Weir. As you'll see, it sort of weaves across the countryside. It's a smallish creek, makes a turn there just before Catamatite, and then through Namurk and Nathalia and enters the Murray River just upstream of Barmer. So that's the site, effectively, that we're working on. Most of the work we've actually done, most of the work I'll actually talk about is the lower section, so from Nathalia downstream. That's roughly where we are. Now, the Broken Creek itself was turned, or I guess agriculture came to the Broken Creek in the late 1800s. So it was recognised as a really good place to graze animals, to grow crops, that sort of thing. The only problem was the late 1800s, the Broken Creek back then was an ephemeral stream. So there was not really much of a consistent flow through the system, and the farming communities had to rely on just occasional water. So modelling sort of suggests that in its earliest days, it probably got really good flows maybe once every 3 to 5 years. So in its earliest days, it was an ephemeral stream. It existed as a series of pools. And so agriculture was sort of set up around that kind of time, but when it was recognised that they needed a better source of water, that's actually when Casey's Weir was built. And the idea, one of the main ideas behind Casey's Weir, was to divert water from the Broken River around through into the Broken Creek. Since then, there's been a whole lot of other modifications go on into the system, and now the Broken Creek is highly regulated. It receives regulated flows through a couple of flows through the Goulburn irrigation system and also through the Murray system, but basically it's a highly regulated system. Harking back to at least a couple of the earlier days, there's one farmer who still has retained the ancient art of haystack building, and these haystacks have been sitting on one of the farms right next to the creek, and so it's really quite an interesting sort of place to be. What actually happened with the Weirpool system is, if we look at the lower section of the river, so if you sort of think of that original picture I'm showing, the top left there, where the Broken Creek has come through Nathalia, it weaves frantically across the countryside before it hits the Murray River. 7 weir pools were put in the earliest days to regulate water, to maintain water flow. 7 small weir pools were put in. The system was basically built up to allow stock and domestic supply regular flow for the irrigation in that area. So most of the work that's going on, and most of the interest is in this lower section where we have seven of the weir pools. So what does the Broken Creek look like? If you look at the Broken Creek, pretty much just downstream of Nathalia, that's a picture of just standing at one of the bridges looking down. It's most of the riparian vegetation in this section's been cleared. It's not a terribly large water body, maybe 20 metres wide, a metre or so deep at the upper sections. The sections just immediately downstream of Nathalia. By the time you get to the middle reaches, things start to look a little bit different. With the highly regulated flow and the high maintenance of flow for a large period of time, you find actually quite a few dead trees within the system, so there's probably a waterlogging issue there. But there are sections where there is a fair amount of vegetation and riparian vegetation in the creek. Still one or two other interesting installations as you drive further down the creek. But you come to a series of weir pools. Now, the original weirs were put in old-fashioned dirt mound style with wooden boards. In 1997, the whole series of weir pools were actually changed. And this particular weir pool here is Kennedy's weir. What actually happened then is the wooden structures were removed, concrete structures, new structures were put in to improve the efficiencies of the water. But most importantly is that fish passages were put on all the weir pools. So in this particular case here at Kennedy's, the flow is from this side going through, and this is actually the top end of one of the fish ladders. So all of the lower section now of the weir, and all the weirs that are actually in the creek have fish ladders to allow fish movement. So that's the same weir pool looking from the other side. By the time you get to the bottom, the last weir pool is Rice's weir, and that's where we've actually done most of our work, because that's I guess where most of the negative or problematic side of the creek have actually occurred. So by the time you get to the lowest weir pool, you're talking about a wall maybe just over a metre high, and it's holding back a fair amount of water right at the bottom of the creek. It's also got a fish ladder, and if the stick just matches it, so the inlet's over the other side of the wall, and downstream of the side you can just sort of see a little bit in there where the fish passage can actually happen. And so standing right on Rice's weir wall looking back, that's what the bottom of the section of Rice's weir looks like. So by now it's quite a wide but reasonably slow flowing and not terribly deep creek. So that's a little bit of the background of what the creek looks like. So a couple of things sort of triggered major events and us getting involved. And the first happened in 2002, and that's when a major fish death incident occurred on the Lower Broken Creek. And something like 100 or so large cod were killed, or large cod died. Created a huge public interest, a lot of conversations about what was going on. But at the time, like most of the fish death incidents, people saw the fish and then a couple of days later got together and tried to work out, by the time they went to the actual creek to study maybe and work out what was actually going on, often the conditions had changed or changed drastically. So while a lot of people had various ideas on what was going on with the fish populations and the fish deaths, people weren't really certain. But anyway, it sparked a lot of interest, certainly when you had a large number and as you can see, pretty big fish actually dying. So that was a major event, a major trigger on what was going on in the management of the system. The other thing that actually people have observed, particularly with the Broken Creek, is the prevalence growth of Azolla. So Azolla is an aquatic fern, grows on the surface of waters, and one of the peaks, which are quite impressive photos in a roundabout sort of way, is, and by 2005 they had sort of one of the worst or the biggest blooms of Azolla on the creek. And so you can see a picture there at the top left, a couple of the guys standing on that weir pool where I showed you just before. In fact, the Azolla is banked up, and it's a good metre thick, sitting behind that wall. And you can see on the top right hand there, just at the probably the exit, I think, of one of the fishways, a great pile of Azolla. The bottom picture is just a standing by the bank along Broken Creek, just to give you an indication of the complete coverage. So in fact, right from Rice's Weir all the way back almost to Nathalia. So look, in a straight line you're talking 30k, so if you convert that into actual creek kilometres, probably a huge distance of creek completely covered with Azolla. So that sort of sparked a fair bit of interest as well. So people then started at the CMA, and people who are interested in managing the system were kind of working out, well, what's the problem here? What's the water quality issue? What's happened with the fish? Are the Azolla playing a part? How does the whole thing function? One other observation, and this is an aerial picture looking back down on Rice's Weir. So the wall is up in here. There's the wall, and this is upstream, and so you can see a patch of Azolla in here. One of the other things that was observed is that often Azolla would kind of crop up. It varied slightly, but somewhere between August and September is when they'd start to see it growing. It would grow massive amounts, but then it would often die, sometimes in December, sometimes January, but there was a period where it really, really proliferated, and then for some reason it would die. So all of these pieces of information were packaged up with the guys at the CMA. They were really interested to try and understand what was going on with Broken Creek, and particularly in Rice's Weir. And that's where we got involved. So at the earliest days, we were driven partly by what the CMA wanted, and then also with us suggesting other things that really would be important to look at if we wanted to understand the overall ecology and how we actually thought Rice's Weir was functioning, what was going on, and then maybe at least understand what you would have to do to try and fix the problem. So what we did was we went into the creek. We were interested in finding stuff about Azolla. How much of it's there? When does it grow? How much of it grows? The rates it grows? And if we could, find out when it died, and if we could, ultimately work out why it might have died, why we have these boom and bust things. So we also wanted to know something about the phytoplankton, the algae in the water column. We were pretty interested in that because we felt that that would play a part in the overall ecology. Generally, nutrients and general water quality were clearly going to be important, particularly dissolved oxygen. So that was one of the big triggers. But we sort of also had a hunch in the earliest days when we were looking at the system that we thought the sediments were going to be a big part of the story, just by looking at and getting a gut feel. So we said to these guys, well, we really also need to get a good picture on what's going on in the sediments and the decomposition processes. So we went in with these very broad aspects, I guess, and over a particular summer, we had a project that looked at some of this stuff. So just as an aside, for those who don't really know, Azolla, as I mentioned, it's an aquatic fern, native. There are two species. The one on the left, Filiculoides. There's two species, and for those that really are interested in care, the one that you can actually tell them quite easily, usually by the root hairs. So the one species, Filiculoides, has just like a single strand almost as a root hair, and it grows a little bit more randomly versus the other species on the right-hand side, which has this nice pinnate, this nice sort of pointy structure, and you can actually identify them reasonably easily. Bottom left-hand corner, sometimes you'll see Azolla growing as green, other times it can actually be completely red. Not known really why, but it's been suggested that usually some physiological condition might be stressing it or changing it in some way. Bottom right-hand picture is perhaps from a microbial angle is why this plant is particularly interesting, and that is Azolla has a blue-green algal or cyanobacterial symbiont in it that can fix nitrogen. So if you take leaves from Azolla, squish them, put them under a microscope, you look at this kind of thing, and you'll see the plant cells here, but here you'll actually see cyanobacterial cells. And these large cells in here actually are able to fix nitrogen. So you've got a plant that may not necessarily rely on nitrogen from its environment and actually can achieve or obtain some of its nitrogen from the creature that's living within the fronds. So it's an interesting plant. Incidentally, just as a complete aside, if you go and do a literature search on Azolla, you'll find great swathes of information. And what most people are actually looking at is, in fact, Azolla is used as a green fertiliser in some parts of the world, particularly Asia, where it can be grown on a water body. You can kill Azolla, or you can let it die, dig it into the sediments or into the soils, and you have a natural fertiliser. So often it's like a green fertiliser. Nutrients and carbon can be put into the soil. So a quick literature search takes you down an Azolla pathway, but for a different reason. So when we started the project, that particular year, this is Rice's Weir Wall, and as you can see the blanket of Azolla over the surface. At this time of the year it was a predominantly pink colour. What did we do? Well, to do some of the growth work, we put out a set of enclosures where we could remove the Azolla and then perhaps add 1 or 2 plants back, and through doing that we could actually get some estimate of how quickly the Azolla was growing, get some sort of idea on how fast things were actually happening. And you would see this kind of thing where you would put in a chamber and come back whatever period of time and the Azolla inside the chamber would have grown. So we could actually get some measure or estimate of how quickly the things were growing. We did a lot of water quality testing, obviously, because water quality was a big part of it. Azolla harvesting, and that's a picture of Carina, and in the end the only way we could really quite easily harvest it, we used the not then patented spatula method of just scooping it out. It grows as remarkably tight with the fronds actually interlocked in one another, so that was the easiest way to sample the Azolla. We did a lot of work with respiration. There's a picture of one of the guys, that's Shane Perryman, and what he's doing there is he's putting chambers. I don't know how clear that picture is at the top, but he's putting out a series of benthic chambers, which are enclosed chambers, and they allowed us to measure what was going on in the sediment, the sort of respiration, and just sort of right up at the top right-hand picture there, you can just make out perhaps very hazily a chamber sitting in with a dissolved oxygen probe coming out of the top, and we can measure how much oxygen is being consumed by the sediment. So what did we find in that particular summer? Well, one of the things that actually happened is we did map one of the typical sorts of growth periods. So you can see the green line there with the date going along, and basically that particular summer Azolla grew in its normal sort of pattern, sort of coming August, September, October, it started to grow, peak growth period, and then by just into the sort of the second week of December or so, it died. Unusually, there was another regrowth, and it's not really happened that many times in Broken Creek. Once the cycle happens, it seems to do its thing, but another growth period actually happened towards the end. Now, the squiggly line is the continuous temperature. Now, one of the things is most of the people we've been working with sort of suggested, well, temperature. The water gets too hot, and that's the reason for Azolla dying. So we mapped the temperature while that was actually happening, and as you can see, the horizontal blue lines sort of halfway up, that's roughly the optimum temperature for growth. And at the top, there's the red line, and that's the lethal temperature. So one of the things we could at least show in this study here is that the growth temperature really only ever got up to the optimum. It never really reached that sort of lethal temperature. So while we could map the growth period, we could never really attribute, for some reason, heat or water quality, or water temperature or anything being really the driver. But anyway, it set us on the path, and we had a period for when Azolla was growing. So that's the sort of picture you got towards the end of the study period, where Azolla had pretty much died away, so the huge blank had gone. Just a couple of other things we actually had to do, and hopefully that's clear enough in a bright room, but we were interested to find out how much Azolla was there. Now, we did this for a couple of reasons. It was the CMA were really interested to know whether they could actually harvest the Azolla and use it for something else or something like that. So we took some measures, and it's fairly difficult to do with a lot of accuracy, and as you can sort of see, there's a huge spread in the numbers. But if you had a single layer of Azolla growing over the water, per square metre, you might have, sort of an average number was something like 100 grams of Azolla there, but it ranged between, could be small as 20 and up to almost 300 grams. For the times when you might get a couple of layers of Azolla, you could have per square metre ranging somewhere between a couple of 100 grams almost up to 500 grams. So to put that in a perspective, we did a rough calculation of what that would actually translate into. So over the first 600 metres of the actual weir pool itself, if you took those numbers and scaled them up to how big the weir pool is, roughly over 600 metres you'd expect almost 70 tonnes of Azolla sitting on the surface. So that's for 600 metres, bearing in mind when it was its worst it was probably quite a few kilometres long. So there's an interesting number to generate for them, and I guess as an aside, one of the things the CMA was interested in is could they actually use this Azolla as an alternative for other purposes. So while it was a pest for them, could they harvest it and use it as a stock feed? But that's another story. Another piece of interesting information is when you actually start looking at the rate that these things grow, and I've presented them there as a dry weight and as a wet weight, basically because the wet weight is what people would actually deal with if they were going to be using this stuff for a stock feed or a fertiliser or anything. Dry weight gives you a little bit more accuracy on what's there. But I guess the interesting numbers to look at are the sorts of ranges. When you start talking about a wet weight of between, say, 12 grams per square metre up to maybe a couple of 100 grams, to put that into a perspective, if you link that back to how much Azolla is actually also in a square metre, it's basically saying that Azolla is growing at a rate roughly doubling its size every few days. So per square metre, that's how quick the Azolla is going. So you're actually dealing with, if you took an area and scraped it away but didn't get rid of all the Azolla, whatever area you'd cleared, within 2 or 3 days it will be all back again. So a really rapidly growing material with a particularly high biomass. So tracking what was actually going on in Rice's weir, along the top there's a period where I've highlighted roughly the Azolla growing, around about where it died. It doesn't die instantly, so I've got a week or so period where it died, then later on where it actually grew. We actually measured the chlorophyll in the water, so the phytoplankton, the algae in the water column. And okay, so generally the numbers, the take-home message from here is generally the numbers as they track are pretty high. So numbers like somewhere between 40 and 60, 70 for a period of time, they're reasonably high numbers. If you went just to the Murray River, you would find a number maybe like 20. So phytoplankton are fairly high in Rice's weir. Immediately after the Azolla died, there was a massive bloom. So a huge spike in the chlorophyll, and you can see that number there, probably around 250 micrograms per litre of chlorophyll. Big bloom of phytoplankton occurred just after the thing died. The numbers bounce around again a little bit, but hard to take a big take-home message from that. But the take-home message from at least the initial part is that post the Azolla event, we did see a big algal bloom. So something else we measured, I guess what we're interested in now, we're starting to get down into the functional sides of things. So what we looked at is the amount of gross primary production. So it's effectively what the algae are doing, what the algae in the water column are contributing to the production and the respiration and all the functional stuff that's going on. So above the water column, in the water column, you've got algae which can effectively put oxygen into the system, generate new cells, and you can think of that as gross primary production. But sitting behind that, you've got sediment respiration, you've got water column respiration, you've got a whole range of other things going on. And so one of the ways to plot this information is over time we measured the primary production basically and related that to respiration. And other than all but one occasion, respiration was always considerably more than production. So the system was, people referred to as heterotrophic, strongly heterotrophic. It was sucking in or using up more oxygen than it was producing. The only time where that didn't happen, and that in fact was the time when the algal bloom was going on, so that kind of makes sense. So looking at that, it's probably some, just as a visual, it sort of indicates that after the death, things really took off and respiration really raced away. So there was an increase in respiration. So as the number gets more negative, it's basically saying there was more oxygen being consumed. So respiration increased after the Azolla event. So I guess that's an observation that could be made. But we do know, and the data is a little bit weak here, but probably because we don't have a lot of data points, but we also know that there is a temperature function in there. So if we look at respiration on the left-hand panel, we know that was reasonably well correlated with temperature. So as the water got hotter, then respiration, in this case, increased, even though it looks like a negative number, because we're talking about O2 going the opposite direction. Likewise, with primary production, there was a temperature component in there, but the variation, it's not explaining all the variation. So I guess what I'm trying to say here is that even though we have a death of the Azolla and a lot of action happening after that in terms of oxygen consumption, there's probably a component being described a little bit by the temperature because the system was warming up, but it may also have been related to other factors that we didn't measure because we can't really explain the variation. And the Azolla may be part of that story as it died. So kind of getting into, I guess, also now getting into a little bit of the nuts and bolts to explain what we might actually be looking at. We looked at the organic content of the sediments. So we basically are talking about here in the sediment, in the mud, in the bottom of this creek, how much of that material is inorganic and how much is organic, how much is dead material and dead plants. And generally those numbers, I guess they're a slightly higher number. We didn't see a huge increase, which I was kind of thinking we might have seen after the event, a huge increase in the amount of organic material because we know the Azolla died and not all of it was transported. But at least in the second event, soon as the Azolla died, we saw we picked up a large increase in the amount of organic material in the sediments. These numbers represent fairly high organic contents. And I guess one of the other things we also did at the time, just to also get a little bit more of a feel for what the organic material in the sediment looks like, we compared immediately above the wall with immediately below the wall. So these two graphs here, these two plots, they're accumulation of all the data we obtained. So every measurement we ever made on the amount of organic material in the sediment. And in these particular plots, if you're not familiar with them, the dotted line is like the average sort of content. The box represents the data that all fitted in with sort of between the 25 and 75% percentiles. So it's basically saying where most of the data was. The error bar is where all of the data is between 10 and 90% of where it fitted, and the dots are outliers. So really the main message from this was that, as you can see, upstream, so that's upstream of the wall where the Azolla is growing, where it's growing and it's dying. There's some movement we know downstream, but the organic content within the actual weir pool itself can be very variable. So we could find pockets of deposition of organic material where the sediment was almost 30% organic material, and that's very high. So a third of the sediment was actually organic material being broken down and being processed in some pockets. So very, very high variability and very large pockets of organic material within the weir pool itself. So we continued on our path and we measured some water quality, and I'll just go through a couple of things here. We took a whole series of depth profiles to understand the oxygen profiles within the weir pool. And I'm just going to talk about a couple of these today, and you'll see the progression. So what we're doing here and how we're reading these, down the left-hand side, those numbers represent the centimetres below surface. So 0 is the surface. On that left-hand panel, the water column was 1.6 metres deep. Along the x-axis is the dissolved oxygen. So if you look at the left-hand panel, it's basically saying just under the surface, the dissolved oxygen was just under 6 milligrams per litre. It dropped rapidly in the next 20 centimetres, and then there was a small but steady decline. So by the time you got to the bottom, the dissolved oxygen in the water column was maybe only a couple of milligrams per litre. The next panel, just another representative column, and the idea here is that the trends, they'll all vary very slightly because every profile is going to be a little bit different, but overall the message is the same. That 5 to 6 milligrams at the top, a strong gradient, and a couple of milligrams by the time you get to the bottom of the water column. A week later, when just following the Azolla decline, things changed really, really pretty drastically, and you can see there from those two profiles that at the top of the water column, the dissolved oxygen was only 2 milligrams per litre. By the time you got to the bottom, it was less than 1 and probably approaching 0. So the whole water body was anoxic for a period of time. We don't know how long because we were only there weekly, but we know the whole water body was anoxic for a period of time. But if you step forward to the 19th, we're now talking about the period where the algal bloom was actually happening. The oxygen content in the water column had changed completely. Algae in the water column were now having a lot more of a say on what was going on and driving the processes. So you can see there at the surface of the water now the dissolved oxygen's 9, so it's above saturation. So it's algae are really chuffing along doing this stuff. And at the bottom, at the depth, you're still only down to 5 milligrams per litre. So the system was really well oxygenated by now, but a strong gradient, still a strong gradient, even though the profile shows lots of oxygen there. And I think lastly, on the water quality, just a couple of other things we measured. We're particularly interested in the nutrients. So we've got 3 panels here, again, with the growth period, the death period, and the second growth. The top panel is ammonia, nutrient in the water. The middle panel is nitrate, or oxides of nitrogen. The bottom is filterable reactive phosphate, or think of it as phosphate. As soon as we saw the death of the Azolla, there was a huge spike in the ammonia and in the nitrate. Incidentally, the numbers are pretty high, particularly for ammonia. They're pretty high for creek anyway, but you can see going from 20, 30 milligram or micrograms per litre up to 180 just after the death of the Azolla. So the death of the Azolla triggered, or immediately after it, there was a huge pulse of nitrogen in the system. Interestingly, and we don't know why, but the phosphorus peak actually occurred a little bit earlier on in the cycle, so just as the Azolla was starting to die. We don't know why. We talked about that for some length, but we could never really come up with an obvious reason for why there was a shift in the phosphorus. But anyway, the take-home message being those values are really high, but the nitrogen peaks after the Azolla dies. So to recap all of that, we packaged all that information up to present a bit of a story. We could show that the pathway, the normal sort of pathway for Azolla happened that particular summer, but we still couldn't really find a cause for its decline. It wasn't temperature, but we still don't really know. What we do know is that Azolla caused a significant change in the water chemistry. We've got some, not tremendous evidence, but we've got some evidence for an increase in sediment carbon, a little bit conflicting. On one event we saw it, a second event we didn't quite see it. It may be our sampling frequency is not enough to have picked it up, but certainly some evidence for an increase in sediment carbon. We certainly see an increase in sediment oxygen demand or sediment oxygen consumption, but we also know that there is probably a temperature factor in there as well. But the Azolla had some effect. We could also say very high carbon loads. We showed very, very strong oxygen gradients and at times could reach 0. And finally, if you do actually have an Azolla bloom and it ended, then there was also potential, at least in this case, that you could follow with the nutrients, basically triggering an algal bloom. So the implications, I guess, of all of that was that the sediments were having a major role in determining what was going on in Broken Creek. Our initial hunch was right, we think. In fact, we're certain we've got enough evidence to put it on that. It seemed reasonable with what we were doing to put a fish stress really being related to oxygen. So we measured other things like sulfide and sulfur content and the toxicity of ammonia and all those sort of things. Most of those really didn't quite cut it. So as far as the fish death or the fish kills actually happened, we're pretty sure that for most of what we could measure or demonstrate, it was probably the oxygen. We know the oxygen can decline really quickly, but we also know it can come back reasonably quickly depending on the dominance of the particular organisms. But I guess the end point was that there was scope, at least potentially scope, to use environmental water to main the dissolved oxygen, and in fact that's the pathway that the CMA and Golden Murray Water have taken. So they now have improved oxygen monitoring. There are actually now 4 probes which are regularly measuring the oxygen within the system at the top and at the bottom, because we recognise that measuring a DO at the top really wasn't going to give you enough information. So there are a series of probes now which are linked to alert systems. once the dissolved oxygen gets below a certain trigger, then an environmental flow or water allocations can be put down the creek to improve the oxygen. We did some interesting trials at the start with a camera sitting up taking pictures of Azolla to see if we could measure and monitor its tracking in and around the weir pool. Wasn't as successful, but what it did do is it gave us a feel for how much Azolla was there on a much regular interval, because it was very much dependent. If you went there at a weekly field trip, you could get a poor impression of how much Azolla was there and where it was sitting, depending on the prevailing winds. So some in situ camera and regular photography has helped understand a little bit more about that. And as I say, it was linked in with environmental water to improve water quality in the creek. So stepping forward now to 2012 is the Commonwealth Environmental Water Office supply a package of that water that now goes down the creek. And they're really interested to be able to demonstrate or to show what that environmental water is actually doing. Based on our initial studies, we've made a series of recommendations. You can now put water down the system. How well is it working? Does it solve the problem every time? And are there any other issues? So the new project being funded by Commonwealth Environmental Water Office has involved ourselves at MDFRC, and Garth and myself, we've enlisted the help of Jack Butler, who's going to be doing his honours in and around this work in some way. We're still working on some of the details with Susan, and particularly Rob Cook, who's going to help out, because we're going to start looking a little bit more at the biofilm activity that's going on there. Mike Stewardson and crew from Melbourne University are a little bit more interested in the system from a modelling side, and Wayne Coster and crew from ARI in Melbourne looking at the fish. So we've got ourselves to this sort of point where if you look at Broken Creek now, that's a rough schematic of what's going on. We know benthic respiration is the big driver of the oxygen consumption. It's mediated a little bit by phytoplankton. They can be a source of oxygen. Oxygen can make its way through the surface, but the amount of oxygen through the surface that happens will depend on how much Azolla is growing on the surface. So those three things are the big drivers. What we know a lot less about, or we don't know much about, is what sort of vertical mixing is going on in there and the inflow. And that's particularly interesting from this perspective, is that we don't have a good feel for how good a water quality upstream is required to fix Rice's Weir, if you kind of see what I mean. The water is transported down from upstream, but if the poor water quality exists for some kilometres, or has the scope to exist for some kilometres upstream, then shifting a package of water into Rice's Weir might not achieve the immediate effects. So part of what we're trying to do is get a bit more understanding about what's going on with the thermal mixing and how that's going to interplay with a longitudinal movement. So that's some of the new stuff that's actually going to happen, and I've mentioned that. When we started working on this project at the beginning of the year, things were all looking really nice and dandy, but of course in March, everyone here will recall or remember that we had major floods within the Broken Creek catchment, and so that's a picture of Nathalia and the creek just sort of winding its way, a little bit of flood water. If you look at the flood there from the NASA satellite that zooms around the place, so the black there is a water body. Now, Broken Creek would normally be such a thin little line you almost wouldn't see it, but you can sort of see where the arrow's coming in there from Broken Creek. Nathalia is just to the right of that. So going on the scale bar of 10 kilometres, you're talking a fairly wide area of water that was actually flooded. So that black area in and around the Broken Creek there. So a lot of water went down that particular creek. Some people got pretty enthusiastic about that, thinking, excellent, 1 in a 100-year flood, this is going to solve all our problems. They dropped the water in the weir pools a little bit, and they were hoping that the flood waters would come in and sweep away all the organic material, and Broken Creek could return to a nice, happy environment for fish and vegetation and all those sorts of things, and they wouldn't have these problems with algae and Azolla. So that's kind of been the hope of some people. So what we've also had to do now is factor in that sort of thinking against where that project was going. Just one other piece of information, when the floodwaters did come through, so fortunately Rob Cook and Nathan had some dissolved oxygen loggers in the Murray River just downstream of where Broken Creek comes in. So you can sort of see the white line there zigzagging along as it does. As the floodwaters came through Broken Creek, dissolved oxygen in the Murray there plummeted. It reached zero for a period of time as the slug of water came through, and then has come back up again. So Blackwater thing that Kerry and Darren have been working on a lot in the last year or so, you saw that beautifully in the week of flood coming down the Broken Creek. So what that sort of showed was that a lot of carbon, a lot of material was coming down the system. It really was a hypoxic Blackwater event. So I guess one of the interesting things around that was, could we then look at what was going on then with the flood? Could we actually go back, start doing some of our carbon measurements, our sediment respiration measurements, and see whether the flood has actually done what some people would hope, and that's scour out maybe some of the sediments, turn the creek around and point it in a slightly different direction. We've only just started collecting data. The project only started last week, but fortunately we went out a week or two earlier and we have collected some data. And the first thing we really wanted to do is at least have a first bash at doing some organic content measurements. So the first column there is Rice's Wall. So that's sort of in and around roughly where we've been taking our previous measurements in the past. So from all the cores we've taken, the sediment in the top few centimetres, there's still round about 20% of the sediment is organic material. The other three columns represent approximately 300 metre distances from the weir wall, and as you sort of head further up into the weir pool itself. And so those numbers, early days, you don't want to read anything too much into the fact that the middle one's a little bit higher yet, but it's still showing that there is a lot of organic material in the sediment, and that probably up to 800 metres, probably almost a kilometre upstream, we're still seeing something like 15% of the sediment is organic material. I think that what that will translate into is that the sediments haven't really been scoured terribly much. There's possibly been as much picked up and deposited within the Broken Creek, but our suspicion will be that when we come along to this summer, it will be as if the flood never happened, and we'll still be at the same sort of point, which is where I'm kind of at now. We're heading into the future. It's a blank screen, and that's because we're speculating a little bit. As I say, my suspicion is that the floodwaters aren't going to have done much because the floods are so slow. The wall structures were still there. A small amount of water was rolling over the top of them. They've picked up a lot of sediment, potentially a lot of nutrient, put it into the creek. So I don't think that the recent flood's going to be any form of saviour. Just what the rest of the creek looks like, well, I guess we'll have to look in a year's time at the modelling, the thermal stuff, the fish movement, and we'll have a better understanding of what the monitoring strategies, or from the monitoring strategies, what that environmental water has actually meant in terms of overall ecology in the Broken Creek. Thank you.